So today's webinar, we're really excited to feature two PhD candidates, Yan Li and Roberto El Khoury, who will speak to us today on very different topics, so we're excited. This is also a rescheduled seminar from one past that we had some technical difficulties, so hopefully the excitement has been building because I know it'll be worth the wait. So just before we get started, a few kind of administrative announcements. If you have questions during the speakers' talks, you can use either the Q&A function or you can use the chat and direct them to us. My name is Erin McConnell, so I'm one of the OTS webinar coordinators. And Ronald, do you want to introduce yourself?
Oh, sure. My name is Ronald Buizen. I'm co-hosting this webinar series together with Erin.
Exactly. So you'll see us as a familiar face in each of the sessions. Just a reminder that since it is recorded, it's also available after on the website, so you can share it with any of your colleagues who you think will find it interesting as well. So we'll have two speakers. Their time will be divided approximately 25 minutes each. We'll have the entire talk. Then we'll have questions, which will be moderated by myself or Ronald. And then at the end, we have a few more announcements, a reminder that our big conference is coming up, the annual meeting, and there's also plenty of award opportunities to apply for. So we'll speak to that a little bit at the end. All right, so our first speaker today will be Yan Li. So I'll let you take it away.
Thanks. Hi, everyone. My name is Yan Li. I'm the 5th year graduate student in UCLA. I'm working in Dr. Fengguo's lab. And the topic I'm going to talk about is structure-based design of antisense oligonucleotides that inhibit SARS-CoV-2 replication. So I'll start with the mechanisms of antisense oligonucleotides. So ASOs is a class of drugs that can target RNA transcript and induce transcription regulations. It functions through two major mechanisms. The first being RNase H1 mediated degradation. The second being steric blocking the splice switching mechanisms. The ASOs are modified to increase the binding specificity and affinity and increase the nuclear stability and pharmacokinetics. These modifications can happen on the base, on the phosphodiester linkage, and on the sugar. Other than these chemical modifications, facilitated delivery systems such as lipid and peptide can be used to further enhance the deliveries of ASOs. Currently, there has been 9 ASOs approved covering disease from neurodegenerative disease to viral infections and also Duchenne muscular dystrophy. These ASOs are chemically modified and have different ways of administrations. So why are we designing a structure-based ASO method? So if we take a look at the drawing of the mechanism of the ASO, you can see that the traditional ASOs bind to the target through full Wilson-Crick pairing patterns. But in a recent paper published by Vincent and Kieft, they stress that each RNA molecule is not single-stranded in the row space. It is actually a compactly folded dynamic structure. And within the RNAs, there are extensive base stacking interactions that is shaping the overall structure of the RNAs. And also, these structures has functional implications. So, for example, if an ASO, a traditional ASO, 2D ASO, is targeting a single-stranded loop of a stem loop, you can think of it as something that is achievable if you only saw the 2D drawings. But in reality, the opening of the stem is 11 angstroms, whereas the paired loop will span a distance of 56 angstroms, and this is clearly incompatible. So we want to stress this problem through structure-based ASO design. As a result, the first part of my talk will be centered around development of 3D ASO design methods. How to achieve 3D ASO design? We bear two principles in mind. The first being that we want to engineer tertiary stabilizing interactions into the ASO and hairpin windings. The second one being we want to achieve both sequence and structure recognition between ASO and the target. Our design templates are based on existing RNA structures. We study our project by inspecting existing RNA structures, and then we design ASO and target constructs based on these RNA sequences. And then after that, we got these ASOs and targets synthesized and characterize the binding interactions, specifically binding affinities, through biochemical methods. And later, we used cellular assays to test the regulatory activities of these ASOs. And finally, we used structural biology methods to determine the ASO and target complex structure. And all the information we got from these characterization steps will be further used as guide for designing ASO designs. And lastly, we're planning to move the ASOs we designed into disease animal model tests. So hopefully the ASOs would be the potential for future pharmaceutical developments. And we call these principles, design templates, and workflows as our structure-based ASO design platform. The RNA structures we inspected are pseudonode RNA structures. And in protein data bank, there are 93 structures in total. And there are extensive tertiary interactions that are stabilizing the structure of pseudonode RNAs. These interactions being major group-based triples, minor group-based triples, Hookersteen interactions, base stacking interactions, and Watson-Crick pairing interactions. The reason why we're choosing pseudonode RNAs is because if you select the pseudonode part of the RNA and eliminate part of the loop, you immediately get one ASO targeting a stem loop structure. And with one pseudonode, you can have two constructs by eliminating different residues in the loop. So this is the case for human telomerase RNA P2BP3 pseudonode. This RNA structure is published by Dr. Julie Fagan's group in UCLA at 2005. And as you can see, this is a typical ASO and hairpin design from this RNA pseudonode. And what's interesting in this structure is that there are extensive tertiary interactions, and it is actually 3 layers. There are 3 major group-based triples, being all U-A-U pairs, and 2 minor group-based triples, being A-G-C pair and A-U-A pair, and 1 U-A Hookersteen interactions. So then we used isothermal titration calorimetry method to measure the binding affinities of the two ASO and hairpin designs we extracted from this pseudoknot. And we found out that their affinities are pretty high. Their KDs are being 57 nanomolar and 34 nanomolar. And we also measured the binding affinity of a positive control, which is ASO2 binding to the ASO2 complement sequence, in which all the residues are binding with Watson-Crick pairs. And we found out that the affinity of this positive control is very close to our ASO2 and hairpin 2 design. This means that the extra tertiary interactions are contributing to the binding affinities to the same extent as those extra UA pairs in the positive control group. And this can also be equivalent to real 3D ASO designs. Our ASOs, 3D ASOs can achieve similar affinities as those 2D ASOs. So secondly, we want to know if the tertiary interactions are contributing to the binding strength. To achieve that, we use mutagenic assays to either mutate the residues or deleting the residues to eliminate part of the tertiary interactions. And we found out that in both templates, if you eliminate part of the tertiary interactions, it will result in a dramatic increase of KD values, which means a dramatic decrease in binding affinities. So this means that all these tertiary interactions are important for the binding affinities. And other than these facts, we don't want to limit our application of 3D ASOs to target only the loop structures. So we re-engineered the hairpins by eliminating the residues that is not in the tertiary interaction module and reconnect the stem regions. And we found out that these our ASOs can still bind to the reconnected hairpins quite strongly. Their KD being 14 nanomolar and 121 nanomolar. And this fact shows that ASOs can be used to target flanking regions of a stem as well. So these two templates extended our application of 3D ASOs. Other than the existing base triple interactions in the wild type human telomerase RNAs, we want to expand our choices of residues when designing 3D ASOs on other targets. So we selected the major and minor group base triples from a summary by Abu et al. In 2011. We incorporated these base triples into the ASO and hairpin design and measured their binding affinities. And also we did a similar thing for the Hochstein pairs as well. I can see that some of these base triples can be fully incorporated into the 3D ASOs without decreasing the binding affinities. And this greatly increased our choices of residues. And however, due to the confidential reasons, we couldn't disclose the details at this stage. So to summarize our 3D ASO design methods, we think that it is key to reconstitute the tertiary interaction patterns of human telomerase RNA into the 3D ASO design. And this tertiary interaction pattern will feature 3 layers of minor Hochstein and major group base triples. And there are 4 templates for us to model our design to target regions both in the loop and in the flanking regions. as we're developing our project, the SARS-CoV-2 pandemic spread globally. So we think that this might be a chance to see if 3D assays can be applied to real life cases. So the SARS-CoV-2 is the positive sense single-stranded RNA virus, and it has a long genome. And there are two critical regions that we're interested. The first being the transcription regulatory sequence, that is leader sequence, that is in the 5' UTR. And the second being frame shifting stimulation element in between the open frame 1A and 1B. The transcription regulatory sequence is controlling the subgenomic RNA synthesis, whereas the frame shifting stimulation element is regulating the expression ratios between the open reading frame 1A and open reading frame 1B. Our 3D assays are SPD1 and PRF3Ps to these two regions respectively. The detailed reason for designing ASO targeting frame shifting stimulation element is that there has been previous success using peptide nucleic acid to target the to target the frame shifting element of SARS-CoV-1. And we did a blast search and found out that the frame shifting element in SARS-CoV-2 only differ from this frame shifting stimulation element of SARS-CoV-1 by 1 nucleotide. So as a result, we designed our 3D assays to target the same 3' region of the frame shifting stimulation element. And this is our design. We targeted the same region, but we engineered tertiary interactions and Hooke's basically the Hooke's interaction and major group base triples into the ASO design. The detailed reason for targeting TRS region is also the same. There has been previous success using PMOs to target the TRS region of SARS-CoV-1. And in the previous results by Newman et al., they designed their 2D ASO based on the prediction that the target is adopting this stem loop structure. And their 3D ASOs is targeting the single-stranded loop regions. So with this model in mind, we designed our first structure-based design on TRS of SARS-CoV-2. And as you can see that the TRS of SARS-CoV-2 is also conserved, as it only differs by 1 residue from SARS-CoV-1 TRS. However, as more research going on, there is gradually a consensus on the SARS-CoV-2 TRS structure, which is different from the previously proposed model. We found out that the SPD1 could still form tertiary interactions with this new structure. However, there's a compromise that there is a TU non-canonical base pair, and we don't know how it will affect the binding affinities at that stage. Other than the 3D ASOs, we designed another ASO called 1.5, which is a 2D design. It targets the same region as the SPD1, but without the tertiary interactions. And the residues are extended to fully disrupt the stem loop 3 in the TRS region. And all the ASOs, including both the 2D ASOs and the 3D ASOs, are ordered as phosphorodiamidate of linos due to its clinical safety and stability. We used the 3D ASOs, SPD1 and PRF3P, and 2D ASOs, TRS2 and SAT1.5, for our viral inhibition assays. In collaboration with Dr. Vati Arumugaswamy's group here in UCLA, we transfected the Hectona-3 cells with these two receptors being expressed with these SOs and incubated them for 24 hours. And after that, the viral infection assays were carried out by the Vati's group. And 24 hours, cells were fixed and cells were stained with immunocytochemistry procedures. And further, the end transcripts of the RNA of the SARS-CoV-2 RNAs are quantified using quantitative RT-PCRs. In the first screening immunocytochemistry experiment, we used the DAPI to stain for cell nuclei, as is shown in the blue signal, and we used the NR616 dye to stain the SARS-CoV-2 spike protein, as shown in the red signal. As you can see that the two DSOs, SPD1 and PF3P, shows the most significant inhibition of the SARS-CoV-2 spike protein signal, comparing with the 2DSO groups and the control groups. And the quantification of the immunochemistry result draws the same conclusion. And the quantitative RT-PCR for the SARS-CoV-2 end transcript give us a more detailed and precise result, but also the same conclusion. So as in this experiment, we only have data points for two concentrations. So we're wondering what's the detailed inhibitory activity profile of these 3DSOs. So we perform the viral inhibition assay again. And this time we perform the experiment as 6 data points, and we quantify the inhibitory activities using quantitative RT-PCRs. And we found out that these two SOs have dose-dependent inhibitions, and their EC50s differ. The EC50 for PF3P is 2.7 micromolar, whereas the EC50 for SPD1 is 0.7 micromolar. And as the ASOs may have potential cytotoxicity problems, and it might affect our evaluation of the inhibitory activities, because if the ASOs are toxic to the cell and resulting in the cell death, it's also resulting in a decrease in the overall amount of the viral RNAs as well. So we used the MTT cell viability assays to profile the cytotoxicities of these two SOs. We found out that the SPD1 have very mild cytotoxicities, whereas the PF3P shows cytotoxicity at 10 micromolar and shows stronger cytotoxicity at 20 micromolar. However, at these two concentrations, the PF3P has already reached over 80% of inhibition. So we think that the majority of the inhibitory activities come from the activity of these ASOs against the SARS-CoV-2 genome. To address the cytotoxicity problems of PF3P, we did a blast search and of PF3P against the human genome, and found out that the PF3P may have off-target bindings with the glucose phosphate isomerase gene in the human genome. So we designed another ASO called PF3P delta 2, in which the first two residues at the 5' site are eliminated, so that we can have less specific bindings with that gene in the human genome. And we found out that these ASO have similar dose-dependent inhibition activities as the PRF3P, and this ASO doesn't have cytotoxicities at all concentrations. So we think that this is an improvement, and also it shows the reproducibilities of 3D ASO design in this case. And also we want to improve the SPD1 design as well. We did that by changing the compromised TU pair to an AU pair, and I found out that this new SPD1 T15A ASO also have this dose-dependent inhibition, and it doesn't have cytotoxicities at full concentrations. Lastly, we want to know if the tertiary interactions are contributing to the binding affinities. So we designed a dual luciferase assay in which the frame shifting element are engineered into the middle between the renal luciferase and the firefly luciferase. And the PRF3P ASOs would bind to the 3' side of the frame shifting stimulation element and induce an inhibition of the frame shifting so that the translation will stop early at the frame shifting stimulation element and reduce the expression of the firefly luciferase. Other than the PRF3P ASOs, we also tested the PRF3P G19C, in which the Hooke's-Steen interaction is very weak, and this might compromise the whole tertiary interaction modules. And another 2D ASO, which is PRF3P delta G, that is covering the same binding regions. We found out that all these ASOs are having dose-dependent inhibition of the frame shifting activities, but the PRF3P are the strongest inhibitors, with the smallest EC50 being 1 micromolar. And I think this result shows that disrupting the tertiary interactions or destabilizing the tertiary interactions will result in a decrease in the regulatory activities, which means that the tertiary interactions are contributing to the regulatory activities.
Jan, you have 1 minute left, so
okay.
Just do a reminder.
Yeah, I'm wrapping up. So the overall conclusion, we got design templates from existing RNA structures and features in major groove, Hooke's-Steen, and minor groove interaction patterns. And 3D ASOs allow ASOs targeting structural regions. It will increase the binding affinities, and it has been successfully used to inhibit SARS-CoV-2 replications. Lastly, I would like to thank all those who provided help to my project. I would like to thank Dr. Feng Guo for guiding me throughout this project, and my lab members, Dr. Grant Schaffner and Julia Chen, for future collaborations. I'd like to thank my collaborators, Dr. Vaiti Alu-Gaswami, Gustavo Garcia, and Arjit, for working on the viral inhibition assays. I would like to thank Dr. Martin Phillips for IDC technical support, and Dr. Andrew Demiano for advice on off-target searching. And my thesis committee members, Dr. Julie Fagan, Dr. Douglas Black, Dr. William Yang, and Dr. Melody Lee, for giving me advice on this project. And finally, I would like to thank my fundings, Whitcomb Fellowship, and NIH and NSF grant. And the preprint of this paper is available on bioRxiv. And thanks everyone for listening.
Yes, thank you, Jan, for this very clear presentation. So if you have questions for Jan, please use the Q&A or the chat function. I will ask a few short questions now, because we are running out of time. But if there are any other questions, I think Jan is happy to answer them online. So actually my question is, when I try to develop a ASO for a particular target, it's always difficult to predict the RNA structure. So there are all kind of prediction programs. So what I always realize is that it does matter how big the part of RNA is that you feed the machine. So it depends on whether you give them a few hundred nucleotides or 1,000 nucleotides. It predicts another structure. So what kind of prediction program would you recommend? And what is yeah, so that's the question. And how would you do this? How would you predict these secondary structures?
I think currently we put our focus on the reported secondary structures as they are more valid, but we do use m-fold predictions of the unsolved RNA structures. So those can be served as our proposals. And I think those, although these structures might not be correct, the SOs we predicted, we designed on these structures can serve as a pool for their experimental characterizations.
Yeah, I fully agree on this. And then my second question would be, I think what you showed now is, I think in one of your very first slides is that there are there are many more applications, but I think you showed the gap mirrors and the splice modulating ASOs. So does this work for both of them? Because I think for the gap mirrors, you have a lot of choice, right? You can move along an exon or an or whatever you want to go. But for the splice modulating ones, you don't have that much of a choice. So it might even be more important to predict these structures and to have this method.
Yeah, I think the splicing mechanisms are mainly using the just blocking methods. But I think the 3D SO still has an advantage to increase the binding affinities. We're currently working on ASOs with these modifications, and we're determining how these modifications are specifically affecting the 3D modules. That's our next step for this project.
Cool. And then I think it's a comment from Masad, a comment and a question. So nice work, Jan. I guess your ASOs which can target duplex regions can be categorized as TFOs, triplex forming oligomers, or ASO TFOs, chimeras. This opens the possibility to consider modified nucleobases as part of your design, pseudo-C, pseudo-G, for example.
Yeah, I think we do acknowledge there was triplex forming oligomers, but I think what's unique in our project is that we're also using Hooke's interactions as our binding module, and I think the triplex forming oligomers are all triplex in their ASOs, but we're combining both the triplex and the one-centroid pairings as well. And also, I think for the pseudo-U and pseudo-C and pseudo-G, that is what we're going to do in the next stage. We're going to try modified bases in our base triples. We have already tried unmodified bases, but we're going to do that as well. Thanks for this question.
And then one very short answer on this question, the last question from Masad. Do you have situations where the ASO invades the duplex region, displacing one of the paired RNA strands, like PNAs when they bind to duplex DNA?
I think currently when we're designing this, we're avoiding disrupting the duplex regions. I think that will be a problems of competing for different conformations of the target. Yeah, but I think if we have no choice in our design, we're going to use ASOs to disrupt some of the double-stranded regions to fit in our design scheme. Yeah. Okay.
Thank you, Jan, for this excellent presentation. So if there are any questions for Jan, you can still use the chat function, or you can ask him personally. Then we move to the second speaker of today. And the second speaker of today is Roberto El Khoury. And Roberto, the floor or the screen actually is yours.
Hi, I can't start my video because the host has disabled it. I'm going to share my screen in the meantime. Okay.
Does it work now?
Yes, and can you see my screen yet? No.
Yeah, we can see you.
I think now you can.
Presentation mode is yet. Yeah, now it is. Perfect.
Perfect.
So we can also see you now.
Amazing. Okay, let me condense this. Okay. Hi, everyone. I'm Roberto. I'm a final year PhD student in the DEMA group at McGill University. Thank you all for being here. So today I'm going to be sharing a bit about my science, specifically talking about G-quadruplex and i-motif structures and their relationship with telomerase enzyme. And I'll also spend a bit of time towards the end just reflecting on the personal and professional development that I've been fortunate to go through during the course of my PhD and outside of my PhD program, and with some concluding takeaway messages that I hope would be useful to some other junior scientists out there in the audience. So to begin with, I'm Lebanese and Colombian in origin. I was born and raised in Beirut. And for my undergraduate studies, I moved to the UAE to do chemistry at NYU. And my first introduction to research was actually in the Trabolsi group, where I worked on metal organic chemically modified, knots, knotted structures, as you can see here, the trefoil knot. And then I moved to Montreal, where to pursue my PhD program under the supervision of Dr. Dama. And that's where I got to work with non-canonical nucleic acid structures. And here I'm showing you a nucleoside that we like a lot in the Dama group, and it's FANA, the 2'-deoxy, 2'-fluoroarabino nucleotides. And you'll be seeing this a lot. I've used it in both G-quadruplex and i-motif structures that I worked on. So to give you guys a bit of background about G-quadruplexes and i-motifs, so G-quadruplexes come from guanine-rich sequences, and as you can see in this diagram, they consist of stacked tetrads of guanine bases that are hydrogen bonded to one another via Hooke's base pairing. G-quadruplexes, the problem with studying G-quadruplexes is that they are very polymorphic in nature, and the same sequence can adopt an anti-parallel, parallel, or hybrid conformation, depending on several factors. One of this could be the nature of the central cation that stabilizes the structure. Another one could be the nature and the length of the flanking bases. Now, on the other hand, i-motifs come from C-rich DNA sequences. So if you can imagine, if you take, if you on one side you have a strand that is G-rich, where you can form a G-quadruplex, the complementary strand would be C-rich, and you would be able to form an i-motif. And so an i-motif structure is a bit simpler, it's not as polymorphic as G-quadruplexes. So i-motifs consist of intercalated hemiprotonated cytosine-cytosine base pairs. Now, the problem with studying i-motifs is that because they require protonation, they're usually only stable at acidic pH. So it's very difficult to study their interactions with biological enzymes. Now, the story that I'm going to be telling you about today is more about the relationship of these structures with human telomerase. So telomerase is actually overexpressed in 85 to 90% of cancer cells. And it is believed that G-quadruplexes form in the telomeres of human chromosomes in order to inhibit telomerase activity. So G-quadruplexes are considered a very important therapeutic target. And ligands have been designed in order to stabilize these structures and therefore reduce telomerase activity. Now, as I mentioned, G-quadruplexes are very polymorphic, and it wasn't until 2014 when our collaborator, Dr. Brian, her group essentially studied the relationship between telomeric G-quadruplex topology and telomerase activity. And what they showed is that unlike other conformation, other topologies, the parallel one specifically is a substrate of telomerase. So they managed to isolate a bimolecular parallel G-quadruplex, and they showed that telomerase can bind to it, it can unwind it, and then it can extend it by adding, the GGG TTA repeats, and therefore elongating this 3' overhang. So this is very cool. And we've been able to sort of contribute to this research as well, because unlike their bimolecular parallel G-quadruplex, we were capable of isolating a monomeric or a unimolecular parallel telomeric G-quadruplex using FANA-G, this modification that I mentioned earlier. And so essentially by substituting guanines with FANA-G at select positions, we are capable of transforming the G-quadruplex system from something that has multiple topologies to something that is exclusively in the parallel topology. And so we wanted to see if this FANA modified G-quadruplex was also a substrate of telomerase. So we collaborated with Dr. Brian, and her student back then, Aaron, ran this great assay, telomerase activity assay for us. So I'll tell you a bit about how this assay works and what it shows us. So in this first lane right here, we see a control 18GGG linear, so it cannot fold into a G-quadruplex, but it is a regular substrate of telomerase. And you can see the regular +6 additions of the TTA-GGG nucleotides. And so what telomerase does is it has an RNA template, and it uses that template to bind to this linear substrate. And then it adds, there's 3 positions where it can add the GGG TTA repeat. And once it adds this first repeat, the enzyme needs to translocate in order to be able to add the next set of nucleotides. And so it does so very processively, and that's why you can get to elongations that are up to 22 or more additions in length. Now, when you look at the FANA modified G-quadruplex that we've designed, we see that just like the control, it is also properly extended by telomerase. And this contrasts with the unmodified G-quadruplex, which has a stuttering extension activity pattern, indicative of poor extension of telomerase. So these results were very exciting, but in my PhD, I wanted to explore the potential role of i-motifs that could form in the complementary strand and whether they could influence telomerase activity. And so the whole point of this is to try to see if i-motifs like G-quadruplex could be an important therapeutic target. Now, there has been evidence that has shown that i-motifs could form the telomeres of human chromosomes. And there has been a study that showed that, they've shown that this SWNT, the single wall nanotube, can selectively bind to i-motifs, to telomeric i-motifs, and this binding event triggers telomerase inhibition. But nothing has been explored apart from this. Now, as I mentioned before, studying i-motifs at neutral pH that is compatible with biological enzymes is a bit difficult because these structures are stable at acidic pH. But a former member of our group, Dr. Hala Bouhassi, incorporated FANA-C modifications into the telomeric i-motif sequence and other i-motif sequences as well. And she showed that she could stabilize the telomeric i-motif at near neutral pH, giving us a substrate that is compatible with biological enzymes. So as part of my PhD, I wanted to test, the influence of telomeric i-motifs on telomerase activity. So I designed these 2 models to sort of start to model what could happen in the telomere scenario. So essentially I'm trying to do is I'm isolating an i-motif, a G-quadruplex, and the telomerase, and see what happens when I put all these 3 together, and whether the extension of G-quadruplex could either be inhibited or maybe promoted. So in these 2 models, we start off with a side-by-side model where the i-motif and the parallel G-quadruplex, which is FANA modified as well, are brought into close proximity to one another with a duplex handle. And on the other hand, in the offset model, the parallel G-quadruplex and the FANA modified i-motif are further apart, and now the FANA modified i-motif is facing a non-complementary region. As controls, we have the unmodified versions, which cannot fold into i-motifs at pH 7 and therefore remain single-stranded. Now, before we proceeded to do any telomerase activity assays, we wanted to check whether our i-motif structures would remain folded in the conditions in which the assays are run, because up until this point, we had only done so in buffer conditions that promote i-motif formation or are simple enough for i-motif formation at pH 7. So along with my undergrad, Morgan Roman, we designed a threat-based experiment to sort of check whether i-motifs could fold under these assay conditions. And essentially, we designed these constructs in such a way that if the FANA i-motif, if the FANA modified sequence would fold into an i-motif, then the two dyes would be placed into closer proximity to one another, and you would get higher threat. Meanwhile, at pH 7, if you don't have i-motif formation, then the dyes would be placed further apart from one another, and you would see lower threat. So here we see two control scenarios at pH 6 and at pH 8.5. At pH 6, we expect both constructs to fold into an i-motif sequence, and we expect high threat, and that's exactly what we see. And on the other hand, at pH 8.5, we expect neither of them to fold into an i-motif, so they would both remain single-stranded, and we would see low threat, and that's exactly what we see as well. Now at pH 7, what was interesting is that only this FANA modified sequence would fold into an i-motif and display a high threat, and you see that it only starts to unfold at higher temperatures, which is normal because now the i-motif is unfolding into something that is single-stranded. And on the other hand, the unmodified sequence is single-stranded from the very beginning at low temperatures up until the higher temperatures. So now we've confirmed that we can proceed with these telomerase activity assays and be confident that our i-motif is actually folding in these conditions. So now our collaborator, Dr. Brian, ran amazing assays for us that helped us, give the story that I'm about to tell you today. And so going back to our side-by-side models that I've showed before, I've also designed two other controls. One, which is a hairpin, where I'm testing to see if other structures other than an i-motif could have any role in telomerase activity. Another, which is a poly-T, to see if other single strands could have any effect. And so here's a telomerase activity assay with these models right here. And what you see is that only in the case when you have a FANA modified i-motif or where you have the single strand that you see an inhibition in telomerase activity, which is very exciting. And you don't see it with any other scenario. And we see the same effect happening in the offset version, where again, when you have the FANA modified i-motif or the unmodified single strand, you see inhibition of telomerase activity. So this is very exciting, and we started thinking that perhaps as the G-quadruplex is being unwound by the telomerase, maybe the FANA modified i-motif or the C-rich single strand are hybridizing to the G-rich sequence and therefore causing this inhibition of activity and processivity. What we also noticed is that there's a subtle difference in the pattern between the FANA modified i-motif and the C-rich single strand. And you see that there's a certain smearing in the FANA modified i-motif scenario. And this suggested to us that perhaps we had non-templated addition, and therefore that there was two distinct mechanisms by which inhibition was happening. Now we proceeded to do another control where now instead of using pH 7, we were using pH 6. And this is relevant because in this case, we are basically forcing our C-rich single strand to fold into an i-motif. So we expect to see the same kind of pattern in the C-rich sequence versus the FANA modified sequence. And indeed, in both cases, we do see inhibition again, but unlike what we expected, the smearing is only observed for the FANA modified i-motif. So here we start to think, is it perhaps the fact that this i-motif is fluorinated that is causing the smearing effect? We're not very sure at the moment. And something we want to do in the future or in the near future is to test the telomerase activity assay at pH 8.5 when we do not expect to see any i-motif structure and we only expect to see a single strand. And we're curious to see what kind of effect we would see in the lanes. In another assay that I want to discuss today is this control assay, where instead of having a G-quadruplex, we're using the linear GGG. And now the i-motif or the C-rich single strand are not tethered to the complementary G-rich sequence. So they're just floating around in solution. But this assay was important because it's the first time where we actually see a difference between the i-motif lane and the single-stranded lane. In the i-motif lane, for the first time, we don't see any inhibition, in contrast with what we see for the C-rich single strand. And this is telling us that the i-motif doesn't necessarily easily unfold in the presence of this complementary G-quadruplex, or sorry, this complementary linear substrate that does not fall into a G-quadruplex, and perhaps the tethering or the close proximity to the G-quadruplex is important. And this also tells us that perhaps another mechanism is actually taking place when the i-motif is in close proximity with the G-quadruplex that isn't explained simply by hybridization. Lastly, one last assay that I want to tell you about today is this last test that I did, where now I took the telomerase, the telomeric i-motif, but I changed the loops of the sequence to a different kinds of loops. And so it's no longer a telomeric sequence, telomeric i-motif sequence. So in one case, I only changed the loops. And in the other case, I changed the loops, but I also added 2 additional hemiprotonated cytosine-cytosine base pairs. And therefore, I increased the stability of the i-motif and made something that was a bit bulkier. So in terms of nomenclature, I called the one in which I only changed the loops SIML for loop. And there's also the control that would be single-stranded, unmodified, so no FANA C modification. And then there is also the other construct that is the bulkier, more stable one. I call it FINS for stability. And as well, you have the CSSS for the unmodified control. Now, what was interesting is, well, so here we just have an additional poly A and poly C to confirm that, again, the fact that it's single-stranded doesn't have any effect, just like the poly T scenario. But going back to the constructs that I just mentioned to you, it was interesting that we saw that the more stable FANA-modified i-motif does not inhibit telomerase activity, neither does this C-rich single strand that is also more stable. And so that was interesting, and it contrasts with what we saw for the one in which we only changed the loops, where now we see inhibition of processivity, but not to the same extent as before. And so in this case, the inhibition of processivity is taking place after 3 translocation events, while here it's only after 1 translocation event. So this seems to be that the inhibition that we're seeing goes through yet another mechanism that we're not even sure how it takes place. So this is how I wanted to end, this part of my talk. I wanted to sort of conclude by saying that we do have evidence that the telomeric i-motif and the C-rich single strand inhibit telomerase processivity, but it's unclear whether they proceed through the same mechanism or not. And this is something that we want to explore more with additional constructs that we hope will give us a better picture. But ultimately, we think that doing single molecule FRET experiments will lead us to really elucidate what is going on in these scenarios. And we hope to be able to get to that in the future. And to conclude this part of my talk, I'd like to do some acknowledgments. First of all, to my supervisor, Dr. Dama, for his immense support over the years of my PhD. I remember, in the beginning, I had almost like I was losing faith in this project, and just seeing how excited and how motivated Dr. Dama was really inspired me, to proceed, venturing into this unknown with exploring whether i-motifs could have any effect on telomerase activity. And I'm really glad that I stuck to this and I reached this stage of the project. I'd also like to give an acknowledgement to our collaborator, Dr. Tracy Bryan, for her commitment to the project and her all the input that she's putting into trying to figure out what is going on in terms of the role of i-motifs. And I'd also like to give an acknowledgement to my undergrad, Morgan Roman, for all the work we did together in the FRET studies, as well as designing some of the last sequences. And lastly, an acknowledgement to Dr. Hala Abu-Asi. She essentially taught me everything I know about G-quadruplexes and i-motifs. And she started this project, and so I'm very grateful to have had her as a mentor, starting on with the PhD. And I'd like to also give a thank you to the funding agencies. And now I'll proceed very quickly to give a bit about more of a personal note of the personal and professional development that I've had during the course of my PhD, and I think has been very influential and has been really helped me proceed in my PhD. So first of all, I'd like to talk about my involvement as an NGO board member of a nonprofit NGO called Montreal that supports the Arabic-speaking LGBTQ to a plus community of Montreal. So I started off as a volunteer and later made my way to the presidency. I essentially put myself in situations that are completely outside of my comfort zone and outside of my expertise. But I'm very grateful to the rich kind of experiences that I had along the way, and they're very varied experiences. I've interacted with asylum seekers and refugees, and that's very important because many of them, only came to Canada only knowing Arabic, so it was very difficult for them to get settled in the very beginning. Through my leadership roles, I've established partnerships with several organizations across Canada and the Middle East. I've also represented the organization in media interviews. And at some point, as a president, I've had to manage a board of directors. And it was a bit intimidating in the beginning because a lot of them were a lot older than me and a lot more experienced. So it was really interesting to navigate these sort of dynamics. But also, during the course of my PhD, I was also always interested in the business world and trying to understand how science ideas can be translated into a business, a startup or whatever. And so I participated in several experiences throughout, one of which is a mini MBA course offered at McGill University. That is a 1 semester long course that offers introductions to strategy, marketing, finance, and accounting. And so that was very informative, and I got to learn so much that I think would be very useful to me in the future. And it also culminated in a group case project, where I had to work with people this is essentially a mini MBA course for industry professionals, people who are not necessarily in science, just people in different industries, different experiences. There's people who are just starting off, people who are managers. So it was very interesting to be placed in a group with people with all sorts of experiences, and coming from a perspective of just being a researcher in science, I felt very intimidated, but I was able to use the leadership or the teamwork skills that I had acquired during my time at Montreal to sort of navigate this and be able to contribute to the case project. I also, as part of during my sort of endeavors, I also took part of the Quebec Scientific Entrepreneurship Program. So for those of you who are based here in Quebec, I would really encourage you to sign up for this if you're interested. I think it takes place every summer, and it really teaches you how to transform lab ideas into solutions. It asks you to come in with a hypothetical idea, and then it walks you through all the steps that you need to take, all the considerations, the sort of resources that you need to think about in order to transform your idea. And so if you're not in Quebec, I really also encourage you to seek out opportunities in your countries, your respective countries, because this was very valuable to me. And lastly, I also participated in a consulting case competition where it was actually offered by the Graduate Management Consulting Association of McGill. And again, I was placed in a group with people from different backgrounds. And we had 1 week time to sort of learn everything we can about digital marketing. And that essentially led me to interview with a very big consulting firm. I really got pushed into it, not really, I didn't have much of a say. Well, I didn't really apply for it, but I wanted to explore whether this thing that was outside my comfort zone was really suitable as a career option for me or not. And so I went through the interview process, and I got to learn more about what it is like to be a consultant in a general consulting firm. So essentially you would be working on several industries that do not relate to science, but they really seek out PhD candidates for their problem solving skills. And so it was through this experience that I probably learned that consulting was not for me, and that I actually really enjoyed what I was doing, you know, in science, in research, and that helped me see sort of where I fit in this world. So I think I've done a lot of these reflections, you know, as I was talking about these experiences. Through my work with telom, I've developed a lot of emotional intelligence by interacting with people from different walks of life. Through my business sort of dabbling, I've learned to think science versus think business. I also think that it was a lot of self-confidence booster when I put myself in situations where I had to learn a great deal about something that I knew nothing of. And you know, when you're in science, you're always challenged that you always need to learn new things, and it could be a bit scary, you know, to have to learn different things. And so I felt like, okay, if I could learn so much about something that I have no experience in, then I'm pretty sure I could learn a lot about something that I am marginally familiar to in science. And so that was very much of a confidence booster. But importantly, and probably what is relevant to this audience, is that through the experiences that I've had outside of my PhD, I've managed to apply a lot of these skills throughout my PhD, especially, for example, when it came to managing collaborations, being able to, you know, organize the project, moving things forward. And so I'm very grateful to these experiences that I've had and to where I've gotten to. And sort of a couple of key takeaway messages that I want to end with that may sound a bit cliche are, first of all, don't be afraid to step out of your comfort zone. You might end up, especially like if you're young, if this is something for, you know, junior scientists, if you're young and you're curious about something, don't be afraid to, you know, seek it out. And, you know, you might end up learning a lot about yourself, about what you're interested. And like in my case, I end up learning that where I'm at right now is where I want to be. Something else that I wanted to sort of bring up is that perhaps, you know, in academic careers, you're trained to be a lifelong learner. You're always seeking out the next idea, contributing to new knowledge. But I've also learned that there's a great deal of learning to do outside of academia that can really reinforce your career, the way you interact, the way you do your mentorship, the way you interact with other professionals, you know, so that's something else that I would really support and encourage everyone to sort of do. And before I conclude, I know I'm running out of time, but I do want to do a plug-in for a symposium that I'm organizing that is also sponsored by the OTS Local Delivery Program, and it's a Therapeutics Intersect LGBTQ2IA+ Symposium, and I'm organizing it along with a few other colleagues at McGill University. And it's meant to be inclusive and not exclusive, so everyone is welcome to attend. It's happening on August 5th at McGill University, but it's also available virtually, and you can sign up using this QR code or this website right here. And essentially, we've secured keynote speakers, and you might recognize Isabel, who used to be part of the OTS board. And they've essentially had great contributions to chemistry and to therapeutics. There's plenty of exciting things on the agenda. So I would really encourage you to attend. And finally, I can, you know, conclude my talk, and I'd be happy to take any questions.
Great. Well, Roberto, that was wonderful. And if your advice was cliche, it's because it was wonderful advice. And so that was really amazing. Thank you for sharing that part. I know Roberto's also really active on Twitter. So if you have questions, I'm sure he'd be happy to continue the discussion there. But we do have a few talks related or questions related to your talk. So the first one says, great talk, Roberto. structures are also cell cycle dependent. Any idea how to include this in your assays?
That's a very good question. And that's true. They are very cell cycle dependent. And it's a bit tricky, I guess, because telomerase isn't necessarily always expressed, and it's not always expressed in all kinds of cells. So it's usually cells that need to be constantly dividing, you know, that would need to constantly express telomerase, and it's only when it is overexpressed that it really becomes a problem. And because these assays are done in vitro, it's very difficult. I have a difficult time conceiving how we could apply it, you know, to something that is that takes into account cell cycles. But it certainly is something that would be interesting to consider, and I'd be interested to explore that further. And if you have any ideas, that would be really nice as well.
Great. And the last question then says, great talk in both projects, Roberto. Are you planning to test i-motifs unwinding in telomerases assay in 37 degrees too?
That's a great point. And yeah, the reason we run our assays, and that's a good observation. And you notice that we run these assays at 20 degrees because that's where our FANA modified i-motifs are actually folding. We do hope to run this assay at 37 degrees, just as a control to see if we do see other sort of patterns, because we do expect that our FANA modified i-motif would not be able to fold. What will be also interesting is there are other ways to stabilize i-motifs at pH 7. So something that we could explore is, you know, adding PEG or other molecular crowding agents to sort of fold these i-motifs even more, and maybe we can reach stabilization at 37 degrees. But the good thing is that we know for a fact that the fact that we're running our assays at 20 degrees does not perturb telomerase activity. And we've done controls to make sure that it doesn't go affected. So even if we only continue running them at 20 degrees, we're still pretty confident that that we're telling the right kind of story through our work. But thank you for the question.
Great. Well, with that, we'll wrap up. We'd like to thank Roberto and Jan for their amazing presentations. I'd also like to remind you that the 18th annual meeting for the Oligonucleotide Therapeutic Society is October 2nd through 5th at the Hilton Phoenix Resort at the Peak in Phoenix, Arizona. So lots of things to do and see there other than the amazing science. There's also several awards. So if you are a trainee, there are travel grants you can apply for. There's also awards for scientists at every level for excellence in science and also in outreach and patient advocacy. So you can check out the OTS website and see all of the awards there. Just a reminder that the short talk abstracts are also due on July 19th. And then the poster abstract deadline is August 19th. So if you have any questions, you can always reach out to info@oligonucleotidetherapeutics.org. And with that, we hope to see you at the next webinar, which is actually next week. And Dr. Bethany Gray-Powell will talk about, or Powell Gray will talk about her experience transitioning from a postdoc into a faculty position.
Thank you.
All right, everyone, take care. Have a great day.